Good afternoon, everybody. I'm Professor Ben Hall. I'm here from Medical Physics and Biomedical Engineering, and I'm going to be presenting Computational Cancer talk. All the right notes, but not necessarily in the right order. And I want to be telling you today about why mutation order matters in cancer, and I'm going to talk you through why computational science can allow us to understand that ordering and gain insights from that. Now, one of the purposes of today's lecture is an introduction to the new Computational Cancer MSc. So cancer research is being transformed through the application of computational methods. This is having a universal impact on the field. This goes from the level of fundamental biology, where computational work is enabling new insights into how we understand the genetics of cancer, the growth of cancer, and the dynamics of cancer, to clinical impacts in early detection, diagnosis, and treatment. The new Computational Cancer MSc is designed to train you in the fundamental skills you need to contribute to this growing area in both computational science and in cancer biology, with a goal to support you for a wide range of future careers. Now, today's lecture is split in two. I'm first of all going to be giving you a scientific taster lecture, talking about cancer evolution and genetic networks, and specifically going to be talking about how cancer-driving mutations combine in complex ways that shapes the process of cancer development. In particular, I'm going to focus on this concept of different orderings and how they can have different effects on the cancer growth and cancer risk through a combination of evolution, epistasis, and epigenetics. Now, the reason I think this is so exciting and so important is because this ordering and the mechanisms behind this ordering tell us about the important features of cancer development. They reveal parts of the unseen life of the cell that tell us why some cells progress into tumors and some do not. After I finish the scientific components of the talk, I'm going to be switching to talk about the new MSc. I'll give you a brief introduction to that and then open the floor to questions either about the scientific talk or the new MSc. So what I'm going to talk about today is going to start off by talking about how we age and how cancer initiates. I'm going to move on to talking about cancer and the data deluge, the ability we now have to collect massive multimodal data sets from different individuals and how we can use them and how we can tame them to turn into useful insights. I'm going to tell you about how we can use principles and knowledge from biology through network modeling to help us understand mutations in cancer. And finally, I'm going to give you examples of where we might be able to understand either cancer risk or cancer outcomes through network models and data analysis. So to start us off today, I'm going to talk about how we age and how cancer starts. Now, I think it's important when we're thinking about cancer, we reflect on the prevalence of the disease and the wider importance. So these are CRUK figures, and they have been updated recently, but they're very similar. 1 in 2 people will go on across the length of their life to develop a cancer. Of those, 1 in 2 will survive for 10 or more years. In 2014, this led to a significant number of deaths in the UK. Now, this problem is not unique to the UK in any way. This is very much a global problem. Cancer, though, is a disease of cells, and we need to understand cells to understand how the cancer initiates. Tumor cells start in the body as healthy, unmutated cells, carrying out their normal function without any issue. There are 40 trillion cells in the adult human body. So even though cancer is very common on a population level, from a cellular transformation perspective, it's an incredibly rare event. A shrinkingly small number of cells go on to develop into becoming a tumor over a lifetime. And this process is generally driven by mutations, in particular accumulated mutations, mutations to multiple different genes that enable the transformation of the cell and the transition from the cell in the healthy tissue, looking normal, acting normally, to one that becomes a tumor, to one that goes on to metastasize. So how do mutations change cells? Well, cells can change their behavior or their phenotypes in lots of different ways. On the side of the screen here, we have the classic hallmarks of cancer, biological behaviors that drive and enable tumor development. But one thing that's implicit here, but not clear perhaps, is that in order for a cell to persist with a mutation, and in order for a mutation to spread in the tissue by a clonal division, these cells need to have Darwinian fitness. They need to have a positive advantage that allows them to persist and expand in the tissue. And both fitness and function, whether it's cancer-promoting functions or just fitness to allow it to survive, are dependent on context. This context might be the tissue. The tissue may be homeostatic. It may be tolerant to some mutations and some types of growth in ways that prevent certain clones from growing and maybe offer blocks to carcinogenesis. The question, it also might be that the cells themselves are not receptive to certain mutations. So if a cell, if a mutation could promote cancer but is not in itself fit, that cell will have a low chance of surviving and growing, and that will minimize the chance of it developing into a cancer. So that mutation may only work to drive a cancer in the context of a cell that's already fit, a cell that's already expanding. So the cell has to be receptive, the tissue has to be receptive for that to work. To add to the complexity here, mutations don't necessarily cause cancer. One of the transformations that's taken place in the last decade has been the sequencing of healthy aged tissues from human beings. What we find when we look at that sequencing is we find that it is normal for these tissues to be replaced by mutated tissues. By the time we're 40 or 70, we are mosaics of mutated cells that have arisen, both through extrinsic processes, tobacco smoke or UV, but also intrinsic processes. Just the aging process, the process of division, can generate mutations that are able to, if they are fit, to take root and to expand. Part of the reason that, that mutations can be tolerated by the tissue is that different mutations can work in the same way, but differ from one another in important facets. So both P53 and Notch1 mutations are very common in healthy esophagus and in tumors of the esophagus. But crucially, despite the fact that they're common and they share a mechanism for that commonality, they both repress stratification. Crucially, only P53 appears to promote cancer development, whereas Notch1, and I'll talk about this a little later, seems to act as a block to cancer development. So different mutations can work the same way, but they can have distinct functions which may promote or demote cancer. Moreover, one mutation is extremely rarely enough to initiate a cancer. Colorectal cancer is a very well understood system. The Vogelstein model has existed for ages, and it argues that the cancer progresses as mutations in these genes are accumulated. And these 4 mutations can be considered to correspond to a worsening of the state over time, a progression of the cancer. However, slightly more recent data suggested actually it's not just these 4 mutations you have to have, these 4 wrong mutations that drive you to cancer. You have to have them exactly the wrong order if the cancer is to develop. If you have these mutations in alternative order, these pathways may not lead to cancer development. It may be because they're unfit, you may be lethal to the cell even, or they may just lose some of their carcinogenic potential. But either way, in order to develop, for the tumor to successfully develop, you have to have the right events in exactly the wrong order. So how does this work? Well, we know that genes act in networks. Multiple genes coordinate together to control the behavior of the cell and determine how it behaves. These networks can create what is known in biology as epistasis. That is to say, when you have 2 mutations together, the combined effect of the 2 mutations in the same cell is not the simple sum of the 2. You can have a negative epistasis, where the combination is no stronger than one of them, or a positive epistasis, where you see a synergy between the 2 mutations. and which mutations thrive and in what orders can be determined by this epistasis. So we've got a detailed picture there. Mutations, their functional effects, their roles in the cell. But we need to bring it back to how does cancer start? Well, healthy aged tissues and cancers both contain multiple mutations. We need to understand cancer, we need to understand which one of these mutations are important, how they combine. And to do this, we need to ask the question, where is the information we can use to tell us what's happening, to tell us what's important? And that brings us on to the question of data in cancer. Now, I'm going to start off with a slightly old quote. I think it's still a very powerful and relevant one. This is from Sidney Brenner, the Nobel Prize winner. "We are drowning in a sea of data, but we are starved from knowledge. We now have an unprecedented ability to collect data about nature. There is a crisis developing in biology that completely unstructured information does not enhance understanding." And I think this is really very much a motivator here. For me, in some ways, despite the fact that this quote is so old, that we're still living in a data-rich world where our knowledge is more limited. In cancer specifically, we have incredibly rich data now from individual patients. Specifically, we don't have just single data sets from patients, but we have what is known as multimodal data. That is to say, data collected by multiple different investigations of the same tumor in the same individual. These might be clinical features, so features of the person, so symptoms and the like, radiology data, pathology data, or genomic data. And these different data sources that have been collected from the same individual can be put together to draw a detailed picture of that individual's cancer. Multimodal data is very exciting in lots of different ways. Multimodal data offers the potential to generate unique insights from the combination of the different types of data with one another. Now, this is just a cartoon to try to illustrate this, but what we've got here are three cartoons representing some surface, a heat map of some surface to suggest some relationship between the data. And what you can see is each of these individual analyses, whether it's imaging, genomics, or biomarkers, is unimodal. Now, the promise of multimodal data is that if you take these data sets, which individually can only be understood in one particular way, and combine them, in the process of combining them, you can identify new patterns that exist from the relationship between the two data sets or three data sets with one another. That you might be able to draw out a more complex distribution of behaviors that can give you new insights into that data. And indeed, this is how it is used this is how it's starting to be used in the clinic, where we were able to draw together data from radiology, data from pathology, there's H&E staining here, and sequencing to get new insights by training models on all of these diverse data. So we have loads of data, but I started this segment by saying about actually the data is not helping us with knowledge. So what's the problem here? Well, data is not enough. Now, I don't want for a second to be appearing to belittle the advances that have been achieved in data science and AI. There are really substantial new insights that these techniques have generated, and that has to be respected. They have enabled new analysis and new prediction and taught us new things about biology. But data intrinsically has limitations, and I'm going to split these into two categories. One of the limitations of data is intrinsic to data itself. Data can be biased, and data can be limited in terms of the number of people who you can collect that data from. And I'll talk a little about that in detail. A more subtle thing I'll also talk about is mechanistic insight. That is to say, data science can sometimes struggle to give you an explanation of what's going on, how a model works. And that has impacts too. So starting off with the concepts of limitations of data, well, one source of bias in cancer data is that cancer data, tumor data, is inherently collected late. As I've mentioned already, cancers evolve from healthy cells. When we collect data from a tumor, from an individual, we are collecting it late in that evolutionary trajectory. We're taking one sample at the end of a process. Now, the process that they have been through will change the cell and change the cell's environment. They're transforming the way they grow, they're transforming the tissue, they're altering their environment. And so there's a complex dynamic process that's taking place that we can't see into directly, that we're not able to collect the data for. Now, practically what this might do is if we look at the cartoon we've got on the right here, if we took a biopsy from a tumor and we sampled a purple population, practically, you can imagine, it'd be difficult to tell the difference between D, B, and A. If all we could tell is we've collected the purple population, that relationship might not be obvious because we can't see into the past. We can only collect that data that's available. Another fundamental limitation of many data sets is that data sets that have been collected from human beings reflect the people who are able to collect the data. So advanced healthcare data, multimodal data, is going to be predominantly collected in wealthier countries. Countries that have advanced healthcare systems that allow them to collect this information. Even within these countries, inequalities in healthcare can limit access to some of these technologies. This is a data problem because this selection effect limits the models that can be built from it. You're not looking at the whole population. You're not necessarily looking at a representative population. You're looking at a subpopulation who just happen to have access. And because you're looking at that subpopulation, that population may not be representative. And there's a lot of great work that's been done showing that biases in public data sets that reflect where the data can be collected actually limit the quality of the models that can be generated. And if you don't collect it, you can't study it. So one thing so moving on to a slightly different topic, there's a different type of missingness in the data. So when we're talking about the diversity of the data, we're talking about people and subpopulations that are missing from our data. But multimodal data tells you that there are lots of different types of data that have been collected, but it doesn't tell you that it's comprehensive. What do I mean by that? Well, I've talked about this idea that biochemical networks underpin cellular biology. This graph on the left here is a model of cellular metabolism. There are 17,000 different metabolites in the cell. We can connect them all up. We know their interactions. We know how some of them are controlled. Those biochemical networks cannot be interrogated easily in patient data. The parameters that define that model are not easily interrogatable. Underneath the biochemical networks are protein structures, which have a three-dimensional fold and have dynamics. And they control the activity of the proteins that control the biochemical networks. None of these things can be easily measured in people directly. And so these, even when you've got multimodal rich data sets, these kinds of information are unavailable. Finally, the final source of bias I just highlight is that cohort sizes are small. Now, cancer is common, but many cancers, individual cancers, are rare. What this means is that even when we've got our ability to collect multimodal data, we may have a very small number of patients. This is further complicated by the fact that common cancers may also be split into distinct subtypes that reduce their effective cohort sizes. So you might not be considering prostate cancer, but you might be considering one specific subtype. More generally, though sequencing technologies are becoming cheaper, the expense of sequencing multiple samples is also problematic. And can be particularly difficult if you're, for example, studying somatic evolution in healthy tissues, not data from cancer patients, but you're trying to do the fundamental biology of aging, for example, and you need to sequence lots of people. Well, the cost is going to be limiting. So there are lots of different limitations in the data that are implicit and not perhaps obvious. But moving conceptually, the lack of mechanism also is a problem. So what do I mean by mechanism? Well, machine learning, data science approaches have a fundamental transformation that goes from data to model. In this process, the data defines the model. The data and the algorithms that are chosen determine how the model is put together, and this model that is generated ultimately allows you to make predictions in terms of classification or numeric or quantitative predictions from that data. Crucially, there's no explanation in those models that tell you where those insights have come from. This black box problem means that you can have a situation where you've got a model that apparently has a very high predictive power, but is not capable of explaining why it's giving you a recommendation it is. The alternative approach to data science is to look at modeling, mathematical modeling or mechanistic modeling. In this situation, you take the data and you couple it with principles. They can be gene regulatory networks, they can be branching processes, they can be evolutionary models, but some sort of conceptual underpinning that you couple with the data to build mechanistic simulations to say how the cells are growing according to these principles, or how the mutations are spreading according to these principles. You can simulate this out, and you can make a different type of model that can also be predictive, but also can be used to explore dynamic space. This is really important because AI, the kind of insights that AI and data science models can draw are correlative relationships from data. Now again, I can't stress enough that this is still incredibly powerful. To be able to predict classifications accurately, outcomes accurately is important. With large language models, we're seeing the application of this to language, allowing us to automate processes like software development and text generation and text analysis. These are incredibly powerful and useful things potentially. The weaknesses of these statistical methods relates though to this idea that they are correlative. These models sometimes need regular retraining. These models can also contain new biases that reflect either the learning method or features of the training data set, or how the model has been trained. Thinking about mechanisms specifically, evidence-based medicine requires some understanding. If you are going to present a patient with a decision in terms of a treatment pathway, you need to be able to evidence that concept. If you have a model that doesn't allow you to interrogate it, doesn't tell you how it works, this is not going to give the individuals strong confidence that this is the right way of doing it, this is the right way of behaving. If you can't explain it and you are making medical decisions, this creates a real barrier. Finally, mechanistic understanding has the opportunity to create novelty. What do I mean by that? Well, if you've got the principles of how something works, you can explore ideas that haven't been included in it before. And this opens up avenues for generating new therapeutics and new biomarkers. And that's only really achievable by understanding. So, to conclude this segment, how do we surf this data deluge? Well, data is powerful and data is big, but it's also limited and incomplete. Data science in isolation can lack the power of explanation. So how can we combine data with knowledge to find these laws of cancer development? And so now we'll switch to talking about network modeling in cancer. In particular, understanding genetics through network models. So I've already mentioned this idea before of complex networks that control carcinogenesis. So genes talk to one another, and this combination of genes can be understood by a graph describing which gene interacts with other genes and how they communicate. Now, the difficulty we have here, particularly for human systems, is that underlying parameters are unavailable and probably totally inaccessible for many systems. But in cancer, what we do know is we know the end states of the cancer. And we may be able to infer some of the chains of events that lead to it through the use of combination of human data and animal model data. So this enables a type of modeling known as gene regulatory networks, or logical models, or executable models. These models are derived from the field of computer science rather than the fields of conventional mathematics and physics, and they're highly abstract. They're discrete and they're finite. So a classic example of this would be a Boolean network, where every gene in the network is represented by its activity is represented by a 0 or a 1. It doesn't represent the physical or chemical interactions, so it don't need to include the parameter fitting in the same way. and you have much more simple relationships. And this gives it less sensitivity to missing components. A final advantage of this type of approach is that this type of model may be amenable to a class of analysis called formal verification or model checking. Model checking is used in computer science to verify the correctness of software and hardware, and we can use those same approaches here. Now, the advantage of model checking is that it addresses the entirety of state spaces by using mathematical proofs. So in executable models, what you can do is you can use model checking to prove your model has the correct types of behavior and the correct dynamics. If you're thinking about cancer and this concept of rare development from a transition from a healthy cell to a tumor, you can guarantee you can capture that rare event. Similarly, if you know certain pathways are never followed, certain cancer development is not possible, you can also guarantee that from the models. So this use of mathematical proofs is very powerful. Now, I'm not going to talk about this in too much detail, but we have a tool that allows you to do this in a graphical way. Being developed in my group at the moment. This is open source, freely available on the web with tutorials for those who are interested. And the concept behind this is that we present to you a blank canvas onto which you can start off by drawing the cells and the cell networks. Through a drag and drop interface, you can put cells onto the screen. You can introduce genes, proteins, and link them all through their native interactions. Finally, just through analyses that are built into the web server, you can also analyze this all live online. So you can very rapidly build and iterate through different models. So this is a powerful modeling paradigm. So how can we use these models to understand cancer? Well, I'm going to give you two examples to close the session. I'm going to tell you about how one gene, homeobox A9, determines the effect of different mutations in blood cancers. And I'm going to also argue that in aged tissue, epistasis explains why some mutations persist and may suggest an opportunity for identifying new biomarkers for risk. So how do different orders of mutation control cancer outcome? Well, we're focusing here on one particular cancer, myeloproliferative neoplasms. Now, these are a rare blood cancer. In about 10% of patients, we find carry two mutations in genes JAK2 and TET2. Now, where both genes are found together, there's a diversity in clinical outcomes. So individuals who seem to have had the TET2 mutation first present later. Their disease is less dangerous. It's not responsive to certain therapeutics, and it's been argued might be a precursor to other cancer developments like AML. That's radically different from individuals who've had the JAK2 mutation first. Those individuals present earlier. They're at higher risk of thrombotic events, and they're responsive to inhibitors of this same JAK2 protein. So you have different groups of people, different cancers determined by the order of mutations or different cancer profiles. And this is actually illustrated by this diagram. Now, you don't need to know exactly what these bars mean, but there are different types of progenitor cell in the blood. The blue and the red and the yellow. And what you can see in the healthy, when you compare the healthy to the TET2 first and the JAK2 first, you can see that the blue cells expand. The CMP cells have expanded greatly in the TET2 population. Those who've had JAK2 first actually have the MEP or erythroid cells expanding rapidly. So we can see the difference in the behavior, not just in the genetics and the clinical presentation, but also on a cellular level. So how can we start to think about this problem? Well, this work, I should say, was led by a talented student, Laura Talamain, in my group. And what she did was she used the BMA to build a minimal network to identify the principles of what a network would have to look like to retain the knowledge or the memory, as it were, of the order of mutations. What she found was all you needed was one gene downstream of both JAK2 and TET2. The key interaction that determined that this network would work is that the target gene, the hypothesized target gene, activated itself. This was a crucial part of the determination of the dynamics. What this means is, if you have a JAK2 mutation occurring first, indicated on the top line, the JAK2 mutation causes overactivity of JAK2. This overactivity of JAK2 is passed on to the target gene, which gets locked in a hyperactive state. That lock is created by that self-interaction. It becomes self-propelling. And subsequently, the loss of TET2 later has no effect on the target. So the target gene controls the target gene is effectively retaining the memory of what JAK2 has done. If TET2 happens first, the opposite pattern occurs. The target gene loses its activity, and again, due to its self-reinforcing activities, its self-activation, it gets locked in this hypoactive state, and it's no longer sensitive to the second mutation. And this simple explanation gave us an opportunity to start thinking about what this gene could be. And so with this mini network, we were able to search databases for genes that have the correct connections. What genes were related to TET2 and JAK2 in the way that would give us this property? We could combine this with data from public databases from the related disease AML. And we could build out the downstream effectors of these 3 genes and phenotypes. This allowed us to identify the gene homeobox-A9 as a potential switch to store the molecular history of mutation. This gene is uniquely found to be downstream of both TET2 and JAK2. It has the right types of connections, and it has a self-activating loop through a gene called MAZE1. And this, we think, gives us an explanation of how the cells can retain it. This gives us an epigenetic memory of the effective order of mutations. Having started with a mini network, we then, and understanding the fundamental dynamics, we built out to include more genes from the biology relating to all the genes that we're interested in the key biological behaviors. This allowed us to identify, make new predictions on the role and relationships of different genes in that network, including Notch1, which had been underexplored for this particular disease, which we could then go back and validate against public data. So what does this teach us? What does mechanistically this teach us about the order of mutation? Well, the model, having built the model, having seen how the model works, this gives us the opportunity to speculate on the mechanism of mutation order. Homeobox-A9 is a well-understood gene in blood. It's linked to cell viability. And we might be able to understand that viability and that relationship to the clinical presentation. TET2 mutation reduces homeobox-A9 activity. That would be expected to reduce cell viability, so slow growth, and lead to a later presentation of the disease. In contrast, JAK2 increases homeobox-A9 activity, giving you the earlier presentation and the faster growth, but at the same time, it becomes more sensitive to the loss of JAK2, which is helping to prop up the hyperactivity of homeobox-A9. So inhibiting JAK2 logically would reduce viability through its interaction with homeobox-A9. We were able to partially validate this through our collaborator Dave Kent, exploring this. This validation did support this view, but it also revealed a greater complexity in the interactions between homeobox-A9 and TET2, giving us room for doing future work. So that's about cancer outcomes. Can we understand cancer risk in the same way? Well, as I mentioned earlier, healthy tissues remain normal throughout life, but they're filled with mutations. These mutations give cells an advantage, allowing them to persist and expand. But crucially, the risks between different groups of people and their mutational profiles, which mutations they have and what types of mutations, vary. Now, what we have on the right here is healthy skin from aged individuals in Singapore and the UK. You can immediately pick out how much more densely the UK tissue is packed with mutations. This relates obviously to cancer because the risk of these types of skin cancers are higher in the UK. So the UK has a higher mutational burden in the skin, and it also has a higher incidence of skin cancer. Also not shown is the fact that in the UK we have more UV damage in our skin compared to the Singapore. So we can understand we've got two different populations with two different risks, with two different mutational profiles. Well, one thing to bear in mind then is that human age tissue is saturated with these types of mutations. Now, this is from the esophagus, and from this point onwards, I'm going to talk about the squamous esophagus specifically. At age 70, the esophagus is completely overrun by mutations. About 2/3 of the tissue is taken over by cells mutated in Notch1, and about 1/3 of the tissue is taken over by cells mutated in P53. I don't have time to talk through this, but actually work with my close collaborator, Phil Jones at the Sanger, has revealed some really important insights into the genetics of these interactions, the expansion. So both Notch1 mutations and P53 mutations allow for expansion of genes in the unmutated tissue. P53 expands slightly less well than Notch1, but they both have a fitness advantage. But that's where the similarity ends, because P53 is selected for in tumours. It promotes carcinogenesis, whereas Notch1 is selected against in tumours. Moreover, Notch1 clones in the healthy tissue can compete with P53 clones and outcompete them, and actually can eject pre-malignant lesions in the tissue. So Notch1 has, if anything, a tumour protective effect, whereas loss of P53 is promoting tumours. Finally, Notch2, a gene related closest to Notch1, is neutral in the wild type. So in an unmutated tissue, Notch2 cannot spread. But if Notch1 has been lost from the tissue, there's an epistatic interaction between Notch1 and Notch2 that allows Notch2 to spread. So how can we understand this? How can we use this to build a model of the interactions between these three genes and gain new insights? Well, again, we can go back to the BMA and build these logical models describing how the fitness is controlled by different mutations. We can embed this in a model of the tissue, the evolution of the tissue. Now what we're looking at here is the cells that are black are unmutated, cells that are blue have Notch1 mutations, cells that are green have P53 mutations. Cells that are red have Notch2 mutations. What you can see is from an unmutated tissue, you see a spread of both P53 and Notch mutations. Over time, initially, they all grow. They all have opportunities to grow and take over and colonize the tissue. As the tissue becomes saturated, you can start to see the green P53 cells be squeezed out, be outcompeted by the Notch cells. You can also start to see Notch2 appear in pink, as the Notch2 is able to expand in Notch1 background, but not in any other background. And so we can use this dynamics, here rendered as a Muller plot, so we can actually observe how you transition from the unmutated state to expansion of Notch1 and P53, to the regression of P53 in competition with Notch1, and you can barely see it in the diagram, but the creation of Notch2 clones in a P53 background, in a Notch1 background. So what does this model tell us? Well, we know that if we look at the data, cells that have mutations in both P53 and Notch1 in healthy are relatively rare. Now, this is true of our model as well. Our model, though, has a specific prediction. It predicts that the double mutant only expands if P53 has happened first. Why is that? Well, Notch1 is fitter than P53. And when you have both mutations in the same cell, the fitness of the double mutants is the same as the fitness of Notch1. What that means is that if you have a Notch1 clone and a P53 mutation happens, it has no advantage, it has no fitness, so it can't compete with its neighbours and it's lost. In contrast, if you have P53 and then Notch, you still increase your fitness. So those double mutants have an advantage over over their parent cells with P53 mutations. So they can continue to expand and persist in the tissue. So the only cells, the double mutants that are likely to survive to be found are the ones where p53 has occurred first and Notch has occurred second. So what else can we tell from the epistasis? Well, Notch2 only expands if Notch1 happens first. Notch1 is rare in tumours but very common in healthy. Now that frequency might limit its use as a biomarker because you don't know how old those mutations are when you look at the tissue. But Notch2, because of its epistatic interaction with Notch1, gives you opportunity to learn about the history of the tissue. If Notch2 levels are high in an individual, then Notch1 mutations have happened early. And we know that because you need Notch1 to have taken over large parts of the tissue before Notch2 is able to expand. So if we see lots of Notch2, that implies we've had a longer-term Notch1 colonization of the tissue. And because we know that Notch1 competes with p53, this suggests that that longer-term colonization will limit p53 growth opportunities, suggesting that Notch2 may be a useful marker of low risk from cancer because it tells you something about the history of the tissue. So to conclude my scientific talk today, the order of mutations can be determined by gene regulatory networks. They can be used to predict clinical symptoms and sensitivity to therapy and have the potential to help us understand somatic evolution and risk factors for early detection and interception of cancer. So I'll take more questions at the end for those who have them, but now I'm just going to give a very brief introduction to our new Computational Cancer MSc. So one of the things that I hope has come across in my scientific talk is the idea that we're undergoing right now a data revolution in cancer. For individuals who come through the healthcare system, we have new opportunities in terms of multimodal data that's available. We've got pathology, radiology, sequencing, clinical data. But the key question for us to address, the pressure is how can we enable clinical improvements from this data? How can we get better patient outcomes from all this data that we're collecting? How can we integrate this data into clinical workflows? How can we use this data in a standardized way? And how can we work with new data types as they become available? So what we have now is not going to be what we have in 10 years' time or 20 years' time. If we have proteomics, if we have methylomics, if we have transcriptomics routinely, how would we integrate this all? Well, the Computational Cancer MSc teaches cancer through the lens of computational biology, presenting the roots and roles of mathematical analysis in tackling this complex disease. This course is jointly run between my department in engineering, medical physics, and the Cancer Institute. And so you take advantage of both the machine learning and modeling expertise in our department and the advanced cancer knowledge in the Cancer Institute. We teach computational science for early detection, diagnosis, and treatment of cancer. We think about it from the point of development, from aged tissues to metastasis, and we're teaching this all from a perspective of modeling and data analysis. We cover AI, we cover data science, and we cover mathematical modeling. We have options available in AI, radiotherapy, and computational pathology. And this course is very much designed for a broad range of different backgrounds. What do I mean by that? Well, one obvious group of people who we'd like to engage in this course is clinicians, people who want to increase their computational skills and bring that back into the clinic. And this course is designed for that and is designed with clinical scientists like Adrian Flanagan. But it's also designed for interdisciplinary scientists from different types of background. Now, I really strongly believe that whether you're from a mathematical type of background or a computational type of background, you'll benefit from this course because you will gain skills in that area and skills in the biology. You'll learn about the biology to be able to bring those two skills together. But equally, if you're coming to this from a biological, life sciences, medical sciences background, this course will support you and will enable your engagement in the mathematics and engagement in the whole course. So we've been designed to bring everybody together to teach people the key skills they need to work. The rough structure of this course is in term one, you'll focus on core modules in cancer biology taught in the Cancer Institute and creating cancer data and computational modeling of cancer taught in medical physics. Term 2, you'll focus on optional modules. And these can be either in purple in medical physics or in white in the Cancer Institute, depending on whether you want to go into a more mathematical, physical direction, or whether you want to go in a more biological direction. And you can pick and choose which options suit you best. Finally, in term 3, you'll draw your focus to a research project where you'll get to work with the top researchers in UCL on new research projects in this area. Now, UCL is very much the place to be doing this. It is a leader in oncology and a leader in computational biology and maths. And it's all supported by a strategic choice to work on engineering better outcomes for cancer patients. That is to say, by embracing engineering and computation to improve early detection, diagnosis, treatment and management of cancer. As part of this, we run the Computational Cancer Collaboratorium. This is a center focused on bringing together the diverse sets of expertise which exist all across UCL into one place. We support this community with a seminar series with some of the best speakers from the field. And those of you who are interested, you should be able to follow this link and you should be able to watch videos of people who've come to speak in the series. We have an early career researcher support network and careers events, most recently with people from AstraZeneca and Johnson & Johnson. And we have an annual hackathon where you can join in, contribute and solve real problems. Now, it's important to sort of highlight the opportunities you get from this course, not just in terms of learning and science, but also in terms of the career paths you can take from here on. So the MSc in Computational Cancer will support a diverse range of future careers. It will support roles in data and computational science, in research and in industry. It will support people who are interested in going into more computational pathology and pathology type analysis. It will also support people going into healthcare, biotech, or related roles in other disciplines that are very different, like specialisms in venture capital or what have you. So I strongly encourage those of you who are interested to check us out online, to apply. Email me if you have any questions. I'm very happy to field them. Thank you all for your attention today. And if you have any questions, I'd be very happy to field them now. If you have any feedback, actually follow this QR code here. I should say this whole talk has been recorded, so you can also come back and look at all of these links, all the many QR codes I've bombarded you with throughout the talk at a later stage. Thank you all for your attention. Any questions? Bill.
Hello, Ben. Thank you very much. It has been a pleasure seeing you talk because I didn't know much about computational cancer. Now I start to understand a little bit. Can you tell me, from your point of view, where do you think this type of research is going to take us in 10 years' time?
So I think it's very hard, to make 10-year predictions. So the question was, for those who didn't hear it, where's this area going to go in 10 years? So a lot of the time, it's actually quite hard to make 10-year predictions because things can change so rapidly. But I think we can, with really absolute confidence, say that computational science is going to play a fundamental role in cancer research. It's already transformed over the past 10, 20 years the integration of bioinformatics and data science into many biological workflows and work patterns. And that transformation is going to continue to take place. And so I think the domain is massively expanding. I think some of the things that are going to positively affect this are the accumulation of more public data sets. And that's going to add fuel to this fire that's going on. The reason for that is because I work a lot in collaboration with different groups, and these collaborations can be massively productive because I get access to their data and I get access to their expertise. But one of the transitions that's happening and has happened is to make data more publicly available. And with that, you reduce the barriers for people to engage in this type of research because you can develop a biological question and then find the data and subsequently access it and start your analysis immediately. Now, I still always will work in collaboration because that's the type of scientist I am. But I think what we're going to see is we're going to see more integration of computational methods, more data science, and more modeling as we start to try and understand these problems and try to tackle bigger and bigger issues, particularly as the computation becomes more readily available and the data becomes more readily available.
And what is the ideal? You know, is it like an early diagnosis, what we are looking for? Is it a better treatment?
So I think the effect is universal. So I think on one end, we understand the fundamental biology of cancer better. Now, that has the opportunity for discovery. So that better understanding gives us the opportunity to say, well, this is a new biomarker that we can use to stratify risk. Or it gives us the opportunity to say, well, this is a weakness of the cell. This can be targeted by this is a potential drug target, and this is something we can go for. But so that's on the biology side. On the clinical side, though, the data sets that are being generated routinely offer the potential for improved diagnosis, including early diagnosis, so more rapid stratification of people, identification of different therapy routes. Personalized medicine is something that can be enabled by these new data sets. So if we're able to say that these groups of individuals have this particular feature in their multimodal analysis or even a radiological analysis, so therefore we're going to target them for this particular treatment, that's a computationally driven process. You have computational models, mathematical models that will take you into that direction. So I think it's going to be an extremely broad range of things. There are going to be effects on therapy, there are going to be effects on diagnosis, there are going to be effects on early detection. I think there are going to be effects on interception. So early detection usually it talks about the process when the clinical process has started. Interception offers you is talking about the opportunity to catch the disease before it becomes presented. This might be related to screening behaviors, but it also might be new screens that we can start to create to identify those at high risk.
Thank you. That sounds so nice.
Hi.
I have a question about the MSc.
Yes, of course.
So, if you're coming from a life sciences background, what kind of struggles will a student face when they approach mathematical or computational methods? So
whatever background you're coming from, whatever the discipline your background is, you'll be supported in integrating into the course. And the core modules will bring you up to speed so you can engage with that. So there'll be components there to support you. So I think I can tell you this from the MSc perspective. There will be the mechanisms in place in the first term to make sure whether you're coming from a life sciences background and you want more programming experience, you'll get that support in those early stages. If you're coming from a computational sciences background, you'll get the support you need in the biological space as well. So that's the perspective of the MSc. From a personal perspective, I'm from a biology background. My background is originally biochemistry, experimental biochemistry, and this is very long ago because this is pre-PhD. But this is one of the things that motivates me to make sure this course is broadly accessible, because I think that everybody from the different disciplines has something useful and important to contribute, and I want to make sure we can unlock that. Hi.
I wanted to ask about the relationship between computational cancer radiotherapy, because I saw that in the MSc there is an optional module, radiotherapy physics.
Yep.
So I was wondering, is it about obtaining data from radiotherapy, or is it about maybe predicting treatment outcome? So what is the relationship between that?
So the radiotherapy physics can be quite a computational and mathematical discipline within physics, and so it naturally aligns. And so one of the opportunities I thought that was there, and one of the things we included as an option, is that it gives you the opportunity to learn a bit more about the related computational methods that are working there. Now, it is a module that is widely available and you can examine on the course catalogue, but it aligns with improving your knowledge of both the cancer and cancer therapies in that specific domain, and also computational work that takes place in that space.
Okay, thank you.
Hi there.
I'm slightly confused between the two ideas, between network theory and the mechanistic models. Like, do we use both at the same time, or do you use?
Yeah, great question. So the question was, what is the relationship between network models and mechanistic models? So the network models are mechanistic. And what I mean by mechanism here is we are saying that this is the rule of how the genes talk to one another. So we're specifying in the model, we know that P53 talks to Notch. We're saying that Notch talks to Notch 1, Notch 2 talks to Notch 1. And those are the principles, the concepts we've taken from model systems, other different experiments, and brought them together to say, well, this is the rules of how the biology works, and then we explore them in the context. So whatever conclusions we draw can be explainable in terms of those interactions. And that's different from a machine learning type model, where you'd be training something on data, you wouldn't have those same principles, same concepts built into it, and you wouldn't get the explainability in the same way. Thank you. Hi there.
Does machine learning have a place in today's research on the topic, given that explainability is a limiting factor?
Absolutely. I think, so don't misunderstand me. This was a talk about mechanistic modeling, and so I drew out the contrast and the advantages of the different approaches. But machine learning techniques have been incredibly powerful. I think it's fair to say we're exploring the nature of that power, and that machine learning models have some different characteristics from other models, but they're still extremely useful and it's still extremely powerful. So yeah, so don't take that from this. There's going to be machine learning and AI in this course, and I do think it is very important. But there are still values to mechanistic models that are unique, and I think that's what I was exploring in the talk today. Any further questions? No? I'll be around for a bit longer afterwards. Thank you all for your attention today, and yeah, enjoy the rest of your evening.